And the pattern of inheritance of such Mendelian disorders can be traced in a family by the pedigree analysis. The most common and the prevalent Mendelian disorders are hemophilia, cystic fibrosis, sickle cell anemia, color blindness, and phenylketonuria and thalassemia. Mendelian disorders may be dominant or recessive. So by pedigree analysis, one can easily understand whether the trait in question is dominant or recessive. The trait may also be linked to the sex chromosome, as in case of hemophilia and color blindness. It is even then that this X-linked recessive trait shows transmission from carrier female to the so X-linked recessive trait shows transmission from carrier female to male progeny. Autosomal dominant trait, first pedigree and example meiotic dystrophy, and autosomal recessive trait, that is sickle cell anemia, in which both parents are carrier. They are not dark in color, but the offsprings are affected. And in first case, autosomal dominant trait, you see the parents are affected, means capital A and small a and capital A and capital A is affected. And so this is it. Now color blindness. Color blindness, it is a sex-linked recessive disorder due to a defect in either red or green cone of eye, resulting in failure to indiscriminate between present in the X chromosome. So 8% of males are affected and only 0.4% of females are affected. So this is because the genes that lead to red green color blindness are on the X chromosome. So males have only one X chromosome and females have two. So probability is maximum in the male case. Okay, the son of a woman who carries the gene has a 50% chance of being colorblind. The mother is not also colorblind because the gene is recessive. Okay, that means that its effect is suppressed by her matching dominant normal gene. That means that its effect is suppressed by her matching dominant normal gene. Okay. A daughter will not normally be colorblind unless her mother is a carrier and her father is colorblind. Okay. Now hemophilia. So it is also sex-linked recessive disease which shows its transmission from unaffected carrier female to some of the male progeny and has been widely studied. So in this disease, a single protein that is a part of the cascade of proteins involved in the clotting factor of blood is affected. So due to this, in an affected individual, a simple cut will result in non-stop bleeding. So heterozygous female that is carrier for hemophilia may transmit the disease to sons. Okay, so heterozygous female that is carrier, and the possibility of a female becoming hemophilic is extremely rare, because mother of such a female has to be at least a carrier and the father should be hemophilic. That is unavailable in later stage of life. Now the family pedigree of Queen Victoria shows a number of hemophilic descendants, and she was a carrier of this disease. Okay, now sickle cell anemia, it is autism linked recessive trait that can be transmitted from parents to the offspring when both the parents are carrier for the gene or heterozygous, right? Are carrier for the gene for or heterozygous. Okay, the disease is controlled by the single pair of allele, the HBA HBS, and out of 3 genotypes, it has 3 genotypes. Only homozygous individual, that is HBS is affected, that is diseased, and then HBA and HBS, that is heterozygous. The individual appear apparently unaffected, but they are carrier. So these are unaffected. Okay, but these are unaffected, but they are carrier of the disease, as there is 50% probability of transmission of that mutate gene to the progeny. Okay, thus exhibiting sickle cell trait, but it shows the sickle cell trait, right? But this shows the sickle cell trait. Okay, now this defect is caused by like now this defect is caused by like so sickle cell anemia. Okay, so we have studied like, so this effect is caused by the substitution of glutamic acid, that is glue, by valine, that is at the sixth position of the beta globin chain of the hemoglobin molecule. The substitution of amino acid in the globin protein results due to the single base substitution at the sixth codon of beta globin gene from GAG to GOG. So glutamic acid is replaced by the valine, that is GAG to GOG. The mutant hemoglobin molecule undergoes polymerization under low oxygen tension, causing the change in the shape of the RBC from biconcave disc to elongated sickle-like structure. Okay, so phenylketonuria. This inborn error of metabolism. So this inborn error of metabolism is also inherited as the autosomal recessive trait. Okay, the affected individual lacks an enzyme that converts the amino acid phenylalanine into tyrosine. Okay, phenylalanine into tyrosine. Now, as a result, the phenylalanine is accumulated and converted into phenylpyruvic acid and other derivatives. So, accumulation of this in brain results in mental retardation. So, these are also excreted through urine because of its poor absorption by the kidney. So, thalassemia. It is also autosomal link recessive blood disease. So, blood diseases are like thalassemia, sickle cell anemia, and hemophilia. And so this also transmits from parents to the offspring when both the parents are unaffected. That is, so carrier means they are unaffected, that is not showing any kind of trait, but they are showing the trait, right, for the gene. So heterozygous. The defect could be now this defect could be due to either mutation or deletion, which ultimately results in reduced rate of synthesis of one of the globin chains. Okay, results in reduced rate of synthesis of one of the globin chains at alpha and beta chains that make up hemoglobin. So this causes the formation of abnormal hemoglobin molecules resulting into anemia. So this causes the anemia. Okay, now which hemoglobin chain is affected? So if it is like alpha thalassemia, alpha thalassemia, and so alpha thalassemia, like production of alpha globin chain is affected, while beta thalassemia, beta chain is affected. Now alpha thalassemia is controlled by two closely linked genes, that is HbA1. Sorry. Okay, so so anyway, so two closely linked HbA1 and HbA2 on chromosome 60 of each. Okay, so the more genes affected, the more gene affected, so less alpha hemoglobins are produced. So beta thalassemia is controlled by a single gene HBB on chromosome 11. Okay. So thalassemia differs from sickle cell anemia is that the former is quantitative problem of synthesizing too few globin molecules, while the latter is qualitative problem of synthesizing an incorrectly functioning globin. So thalassemia differs from sickle cell anemia in that the former is quantitative problem of synthesizing too few globin molecules, while the latter is qualitative problem of synthesizing an incorrectly functioning globin, right? So thalassemia differs from sickle cell anemia in that the former one is quantitative problem of synthesizing too few globin molecules, while the latter is qualitative problem of synthesizing an incorrectly functioning globin. So chromosomal disorders. So chromosomal disorders on the other hand are caused due to the data absence or excess or abnormal arrangement of one or more chromosomes. The failure of segregation of chromatids during cell division cycle results in the gain or loss of chromosomes called aneuploidy. So aneuploidy and polyploidy. The chromosomal disorders on the other hand are caused due to absence or excess or abnormal arrangement of one or more chromosomes. So failure of segregation of chromatids during cell division cycle results in the gain or loss of chromosomes called aneuploidy. So for example, like Down syndrome results in the gain of extra copy of chromosome 21. Similarly, Turner syndrome results due to loss of an X chromosome in human females. So XO. Okay, so failure of cytokinesis after telophase stage of cell division results in an increase in a whole set of chromosomes in an organism, and this phenomena is known as polyploidy. So failure of cytokinesis after telophase stage of cell division. Cell division results in an increase in a whole set of chromosomes in an organism, and this phenomenon is known as polyploidy. Okay, this condition is often seen in plants. So the total number of chromosomes in a normal human cell is 46, that is 23 pairs. Out of these, 22 pairs are autosomes, and one pair of chromosomes are sex chromosomes. So sometimes, rarely, either an additional copy of a chromosome may be included in an individual, or an individual may lack one of any one pair of chromosomes. Or additional copy of chromosome may be included in an individual. So these situations are known as a trisomy or monosomy. So monosomy is when the one chromosome is lacking, and trisomy is when the one chromosome is getting added. No, such situation leads to a very serious consequence in the individual. So Down syndrome, Turner syndrome, and Klinefelter syndrome. So these are the common examples of chromosomal disorders. So Down syndrome, this cause of this genetic disorder in the presence of an additional copy of chromosome number 21, that is trisomy 21. So Down syndrome, the cause of this genetic disorder is the presence of an additional copy of the chromosome number, that is 21, that is trisomy 21. Now this disorder was first described by Langdon Down in 1866. The affected individual is short stature with small round head. Small round head, okay, furrowed tongue, and partially open mouth. Palm is broad with characteristic palm twitch. Cyclic and psychomotor and mental development is retarded. So physical, psychomotor, and mental development is retarded. Klinefelter syndrome. The genetic is also caused due to the presence of an additional copy of X chromosome, resulting into karyotype of 47. Okay, so XXY. So this genetic disorder is also caused due to the presence of an additional copy of X chromosome, resulting into karyotype of 47, that is XXY. It means 44 plus XXY. Such an individual has overall masculine development, however feminine development, that is development of breast, gynecomastia, also expressed. Such males are sterile. Now third disease, chromosomal disease is Turner syndrome. So such a disorder is caused due to the absence of one of the X chromosomes, that is it is 45 with X. So such females are sterile, ovaries are rudimentary, and they lack the other secondary sexual characters. Okay, so in the first diagram you can see the tall stature with feminized character, right? And second is a short stature and underdeveloped feminine character. So short stature and underdeveloped feminine character. The diagram is the representation of genetic disorder due to sex chromosome composition in humans. So these are the genetic disorders due to sex, due to problems in due to abnormalities in the sex chromosome composition in human beings. So Klinefelter and Turner syndrome related to the sex chromosomes, right? Now Down syndrome is related to autosomal kind of thing. So you can see the flat back of head and a broad flat face and a flat back of head. Many loops on fingertips. Then palm crease. Okay. Big and wrinkled tongue. And then congenital heart disease. So the representative figure showing an individual inflated with Down syndrome and the corresponding chromosome of the individual. In humans, a normal female has 22 pairs of autosomes and a pair of sex chromosomes XX, and male has 22 pairs of autosomes and a pair of sex chromosomes as XY. So in chicken, sex chromosomes in male are like ZZ and in females are ZW. So mutation is defined as change in the genetic material. In Klinefelter syndrome, the condition is XXY. This can be easily studied by analysis of karyotypes. Okay, the micrograph of the red blood cells and the amino acid composition of the relevant proportion of beta chain of hemoglobin from a normal individual and from an individual with sickle cell anemia. GAG, GUG. So you see GAG is replaced by GUG. CTC and CAC. CAC, CTC, and then GAG, right? Then GAG, and here it is GTG.